4 billion years ago, Earth was still a restless and unpredictable world. The oceans were warm, restless, and filled with chemical potential. Above them stretched a dense atmosphere of carbon dioxide, nitrogen, steam, and volcanic gases, glowing gently beneath a young sun whose light struggled to penetrate the thick blanket of clouds. And beneath these clouds, in the shallow pools and mineral-rich shorelines, something new was beginning to take shape, something that would change the direction of the entire planet. For millions of years, chemical reactions have been forming random chains of molecules, linking simple building blocks into longer sequences that stored bits of information. Most were fragile. Most broke apart almost as quickly as they formed, but not all of them. A few rare chains could bend, twist, fold, and perform tiny tasks. They were not alive, not yet, but they were drifting closer to the edge of life with every accidental improvement. These molecules were the ancestors of RNA, and the world they created is known as the RNA world. It is one of the most mysterious, debated, and important chapters in the origin of life. Because in this era, chemistry crossed a threshold. Molecules stopped behaving like simple matter and began behaving like something that competed, adapted, and persisted. They began copying themselves. They began evolving. And from these countless cycles of survival and failure, the first whisper of tree biology emerged. To understand how this happened, we must imagine the Earth as it was then. Not a violent inferno constantly spewing lava, but a planet with long stretches of calm. The surface was dotted with volcanic islands, rocky shorelines, tidal pools that filled and evaporated, and warm ponds that concentrated chemical ingredients over long periods of time. Rain fell constantly. Lightning cracked through the air. Winds drove waves that stirred the oceans, and beneath the surface, hydrothermal vents pumped out minerals and gases into the dark water, feeding chemistry that would eventually lead to organization. During this time, nucleotides, the molecules that would eventually form RNA, were becoming more common. They accumulated in water, clung to mineral surfaces, and occasionally joined into long strands. These strands twisted into shapes that allowed them to perform simple functions. Some attracted other molecules, some folded into tight spirals. Some even behaved like tiny catalysts, helping reactions happen faster. These early catalytic strands are known today as ribozymes, and they are one of the most important clues supporting the RNA world theory. The earliest ribozymes were probably not elegant or efficient. They were clumsy, unstable, and rarely effective. But in a world where chemistry repeated endlessly and millions of new molecules formed every hour, even one ribozyme with slightly better stability or slightly better structure could outlast the others. It could persist a little longer. It could influence its surroundings differently. And slowly, across thousands of generations of molecules, the population of surviving strands shifted. The world was selecting molecules without even knowing it. But replication, true copying, was not yet happening reliably. Most RNA-like chains fell apart before they could copy themselves. Others copied only fragments. The environment constantly destroyed them. UV radiation, temperature swings, drying cycles, and waves repeatedly tore apart the fragile chains. But the very forces that destroyed them also helped create new ones. Drying caused condensation reactions. Sunlight powered chemical bonds. Tides concentrated building blocks. And so, despite destruction, more strands always formed. Then at some point, a critical milestone was reached. A strand appeared that could help assemble a copy of itself. Not perfectly, not consistently, but enough to matter. It might have acted by attracting nucleotides into positions that matched its own sequence, or by folding into a shape that stabilized partial copies, or by serving as a template for complementary molecules. However it happened, this strand crossed an evolutionary threshold: inheritance. For the first time in Earth's history, a molecule existed that could pass its structure to the next generation, and with inheritance came evolution. Copies were never perfect. They contained errors, small differences in sequence that changed the way the strand folded or reacted. Some errors weakened the molecule, others strengthened it. But every error was a possibility. Every mutation was a test, and the environment was the judge. Over thousands of years, this molecular competition intensified. Strands that resisted heat survived longer. Strands that folded faster produced more copies. Strands that catalyzed their own assembly performed better than those that required outside help. The ocean, the tides, the shorelines, and the vents created endless challenges. Those that could not adapt vanished. Those that improved continued on. This gradual process began shaping a world where molecules interacted in increasingly sophisticated ways. Some strands might have stabilized the building blocks around them. Others might have acted like regulators, controlling which reactions happened. As these networks grew, the molecules were no longer isolated chains drifting randomly in water. They were becoming parts of systems, primitive, decentralized, unstable systems, but systems nonetheless. Meanwhile, environmental cycles were playing an essential role. During drying phases, molecules were forced into close contact on mineral surfaces, increasing the chances of forming longer strands. During wet phases, those strands dissolved into water, where they could fold, react, and replicate. Day-night cycles heated and cooled the pools, breaking some strands while allowing others to survive. These cycles, repeated over millions of years, acted like stages in an unwritten experiment, refining molecular behavior step by step. Eventually, certain strands gained the ability to assist not only their own replication, but also the replication of others. This created something remarkable: cooperation. Molecules that supported each other's existence formed mutual relationships. If one strand catalyzed a reaction that helped a second strand assemble, and that second strand then supported the first, a stable partnership could emerge. These molecular alliances represent an early form of biological teamwork, long before cells and long before DNA. But cooperation was only half of the story. Competition also intensified. Some strands interfered with the replication of others. Some broke apart their rivals. Some replicated faster but with too many errors. This created an arms race at a molecular scale. The only strategies available were chemical, but they were enough. Evolution did not require intention. It simply required variation and inheritance, and by this point, both were firmly established. Through these endless cycles, certain strands achieved something extraordinary: catalytic replication. They not only served as templates, but also actively helped chemical reactions occur. These strands were the first replicases, molecules that could accelerate the assembly of complementary strands. With replicases in play, replication became faster, more reliable, and more competitive. Suddenly, the early Earth was filled with a new kind of molecule, evolving RNA-like systems that could spread through environments far more effectively. But RNA alone was not enough to build complexity. It needed compartments, boundaries, and protected spaces where reactions could take place without being diluted by the ocean. Fortunately, nature provided these as well. Fatty acids and lipid molecules naturally form small droplets, micelles, and vesicles in water. These droplets were simple, fragile, and constantly forming and breaking apart, but they could trap RNA-like molecules inside. When that happened, the internal chemistry became different from the outside. The droplets were the earliest protocells, still not alive, but stepping much closer than anything before them. Inside these primitive compartments, RNA strands could accumulate, interact, and evolve more efficiently. Some compartments had better mixtures of molecules. Others fell apart quickly. Over time, those that contained more stable and efficient RNA systems lasted longer. They split and reformed during turbulent conditions, carrying their molecular contents into new environments. With every cycle, the combinations of RNA inside changed, and gradually, the earliest cooperators between RNA and lipids took shape. This was the dawn of molecular ecosystems, complex interacting networks of RNA strands, lipids, and chemical reactants. Nothing was alive yet, not in the way we recognize life today. But everything needed for life's eventual emergence was now present. Replication, variation, competition, cooperation, compartmentalization, and self-sustaining chemical cycles. The RNA world was not a moment. It was an era, long, ancient, and full of experiments. Most experiments failed, but the ones that succeeded formed the blueprint for all future biology. DNA, proteins, cells, metabolism, evolution itself, all trace their origins to this strange, fragile time when molecules first learned to carry information and shape their own future. And as the RNA world matured, something inevitable happened. Chemistry transformed into biology. The first true protocells were about to emerge, and with them, the first living systems. As the RNA world expanded across the planet's tidal pools, coastlines, and shallow seas, new molecular behaviors began to emerge, behaviors more complex and coordinated than anything seen before. The earliest RNA strands were simple, short, and fragile. They performed only one or two functions at best, and those functions were often inefficient. But as generations passed and replication cycles multiplied, strands grew longer. They folded into new shapes. They developed new catalytic abilities, and with every improvement, the boundaries between chemistry and life grew thinner. The Earth itself acted as a vast laboratory. Every environment tested the molecules differently. In hydrothermal vents, heat pulse cycles drove rapid chemical turnover. On tide-washed coasts, drying and rehydration refined the assembly of longer RNA strands. In shallow ponds warmed by sunlight, UV light destroyed many strands but also created more reactive chemical fragments that formed new ones. These varied environments ensured that no single type of molecule dominated. Instead, countless variations existed simultaneously, each shaped by the conditions around it. Some of these RNA strands began to achieve remarkable stability. Their structures folded in ways that protected their cores from hydrolysis and heat. Others developed catalytic properties that helped them join nucleotides into chains or cut apart other strands. These catalytic molecules introduced a new form of chemical intelligence. They behaved almost like tools, shaping their environment and influencing chemical flows around them. And where many tools coexisted, more complex networks could form. In certain locations, the conditions favored longer, more complex RNA structures. Thick mats of minerals rich in borate, phosphate, and metals bound nucleotides and stabilized chains long enough for them to interact. Sometimes these chains overlapped, creating hybrid strands that carried pieces of information from multiple ancestors. These hybrids could fold differently, act differently, or replicate differently. Variation increased, and with it, the chances of discovering new catalytic abilities increased as well. Eventually, a strange kind of organization appeared, networks of RNA molecules that interacted with one another in mutually supportive ways. One strand might catalyze the formation of another. That second strand might help stabilize the first. A third might assist in the breakdown of waste molecules that interfered with their function. These interactions formed cycles, primitive metabolic loops powered by environmental energy and maintained by chemical cooperation. The cycles were fragile, temporary, and easily disrupted, but they represented something new, sustained molecular order. Competition was still fierce. Most RNA systems failed quickly. Even the best ones faced constant threats. Waves washed them away. Temperature spikes split them apart. Overly rapid replication produced too many errors, causing strands to lose their structure. The environment was merciless, but this harshness ensured that only the most adaptable strands persisted. Natural selection, though blind and mechanical, began pushing RNA toward greater function and stability. One of the greatest evolutionary pressures came from the balance between accuracy and speed. Molecules that replicated too slowly were destroyed before making enough copies to matter. Molecules that replicated too quickly made too many mistakes, producing copies that were too different to retain their catalytic abilities. The sweet spot lay somewhere in between, a balance that allowed strands to survive and evolve without losing their identity. This balance is still seen in life today. Too much mutation destabilizes a species, and too little prevents adaptation. As this balance was achieved more frequently, replication rates increased across many RNA systems. Strand populations grew, spread, and diversified. each region of Earth became home to slightly different molecular communities. In one pool, high-temperature cycles selected for strands that could withstand heat. In another, minerals favored complex folded structures. In yet another, constant UV bombardment forced molecules to develop protective conformations or pigmentation. Diversity exploded at the molecular level long before life itself diversified. During this booming diversity, one category of RNA molecules rose to special importance: ribozymes that supported replication. These replicase-like molecules folded into shapes that allowed them to help assemble complementary strands. Once formed, they became powerful evolutionary drivers. A replicase could dramatically accelerate the production of new strands, spreading its genetic influence across a pool or compartment. Any protocell that captured a replicase inside its boundaries gained a massive advantage over those that lacked one. But replicases also intensified competition. A poor replicase slowed down a molecular community. A faulty replicase introduced too many errors. A highly efficient replicase helped its own sequences flourish at the expense of others. The presence of replicases, therefore, created complex selective landscapes. Molecular communities evolved strategies to regulate replication, stabilize complementary strands, and manage error rates. Even in this pre-life world, evolutionary dynamics resembled those found in modern ecosystems. Another critical development came from the environmental compartmentalization provided by lipids. Fatty acids and simple membrane-forming molecules naturally assembled into spheres. These tiny vesicles trapped RNA strands inside them. Some vesicles had more RNA than others. Some had none. Others developed membranes that allowed certain molecules in or out. Over time, vesicles with beneficial RNA systems became more stable. They absorbed nutrients from their surroundings more effectively. They grew larger. They divided mechanically under turbulence. The key difference was that the contents of the vesicle mattered. A vesicle containing random chemistry might survive for a few minutes or hours. A vesicle containing a functional RNA system could survive longer, divide more often, and spread more effectively. The environment selected not just molecules, but combinations of molecules within protected spaces. This selection was the foundation of cellular evolution. In some vesicles, RNA strands cooperated to maintain the integrity of the membrane. They produced small molecules that strengthened the lipid layer. Others produced molecules that facilitated the uptake of nutrients from outside. Still others regulated internal chemical reactions by catalyzing or inhibiting specific steps. These were the earliest glimpses of biological regulation. The emergence of such regulation represented a profound shift. Molecules were no longer simply reacting to environmental conditions. They were modifying those conditions. They were shaping their internal environment, making it more favorable for their own survival. This self-modification is one of the hallmarks of life. The RNA world was slowly learning to protect itself. Meanwhile, external pressures continued shaping the evolution of molecular systems. Storms, tides, and temperature shifts caused vesicles to rupture and reform constantly. Each rupture mixed the contents of different vesicles, creating new combinations of RNA. This recombination introduced even greater diversity, leading to molecular systems with entirely new capabilities. Some systems became more robust, others became more efficient. Over many generations, the best combinations outcompeted the rest. One of the most important steps in this period involved the development of ribozymes with multiple functions. Some RNA molecules folded into structures that allowed them to catalyze more than one type of reaction. These multifunctional strands were rare but powerful. They simplified molecular networks by reducing the number of strands needed for a functional chemical ecosystem. They also increased the likelihood that a single vesicle could contain all necessary components for replication and maintenance. As multifunctional ribozymes spread, molecular networks grew increasingly integrated. They formed reaction cycles that supported replication, energy harvesting, and molecular repair. These cycles resembled primitive metabolic systems. They were inefficient and dependent on environmental input, but they could maintain the internal structure of a vesicle long enough to replicate its contents. This was a crucial step toward the emergence of autonomous protocells. Eventually, the RNA world reached a tipping point. Some molecular networks had become so efficient that they could persist even in challenging conditions. They no longer relied solely on chance for assembly. Instead, they used their own catalytic abilities to reproduce key components, repair damaged strands, and maintain stable internal chemistry. These networks behaved like living systems, even though they lacked the complexity of modern life. The seeds of biological organization had been planted. In vesicles scattered across the early Earth, millions of tiny molecular experiments were running simultaneously. Some vesicles failed quickly. Others lasted longer. A few developed the ability to stabilize their internal chemistry through feedback loops. These loops regulated pH, temperature sensitivity, and molecular concentrations. With regulation came control, and with control came resilience. The next step in the evolution of life was approaching. The transition from an RNA-dominated world to a world where proteins and DNA would eventually take over. But before that transition could occur, RNA needed to develop even more advanced capabilities. It needed to become the master of its own replication. It needed to shape the chemistry inside protocells. It needed to push molecular systems toward true autonomy. Millions of years of experimentation lay ahead, but the direction was clear. The RNA world was no longer simply a world of molecules. It was becoming something new, something that could adapt, evolve, and endure. And in the depths of this ancient world, the blueprint for all future life was being written. And so, as the young Earth continued to cool, the oceans deepened, and the chemistry within them grew stranger, richer, more unpredictable. For millions of years, volcanic islands rose and collapsed, hydrothermal vents opened and sealed, and meteors continued to pepper the surface. Each impact stirred the oceans like a cosmic mixing spoon, scattering minerals, metals, and carbon-rich dust. The recipe for life was collecting, piece by piece. But something was still missing, something subtle, something that required not just the right ingredients, but the right stage. That stage appeared in the shallow coastlines, the tidal pools. Here, water levels rose and fell every single day. Here, sunlight could reach the bottom. Here, molecules weren't endlessly diluted in the vast ocean. They were trapped, concentrated, forced to interact. And inside these pools, chaos took charge. When the water evaporated under the young sun, it left behind thin films of chemicals, layers so small you couldn't see them, but so dense that every atom was practically bumping into another. And when the tide rushed back in, the pools filled again, dissolving, mixing, scrambling everything in sight. Evaporation, flooding, heating, cooling, over and over. Every day, every season, every year for millions of years. Nature was running experiments, trillions of them. And in this madness, simple molecules began linking together. Amino acids snapped into chains, sugars formed rings, fatty molecules assembled into tiny bubbles, primitive compartments. And whether you call them droplets or vesicles or protobubbles, these little structures did something incredible. They trapped other molecules inside them. For the first time on Earth, chemistry was happening in a confined space. It wasn't life, not yet, but it was the closest thing the Earth had ever seen. Inside these microscopic bubbles, reactions sped up. Molecules stayed close long enough to interact, combine, or break apart. Some bubbles survived longer than others, especially those reinforced with minerals or more stable lipids. Others popped instantly, releasing their contents back into the pool to start again. Creation, destruction, creation, a cycle as old as the planet itself. Meanwhile, storms raged overhead, lightning shattered the sky, injecting raw energy into the pools. UV radiation poured down, breaking some molecules but powering new ones. Elements from volcanic ash rained into the water, adding iron, nickel, sulfur, metals that would one day power enzymes. Everything was building, every tide, every storm, every boom of distant thunder. The Earth wasn't quieting down, it was preparing, because deep within these shallow pools, something extraordinary was beginning to take shape. A pattern, a tendency, a direction. Molecules weren't just forming randomly anymore. They were starting to behave in ways that made them harder to break apart. They were becoming persistent. They were becoming selective. They were becoming almost organized. And soon, one particular class of molecules would change everything, not by being strong, not by being complex, but by doing something no molecule had done before, something that would define the next 4 billion years of evolution. They would learn how to store information, and that is where everything truly begins. For millions of years, molecules had formed, collided, and broken apart with no memory of what came before. But now, a new kind of molecule appeared, one that could remember its own structure, one that could make copies of itself. It wasn't DNA, not yet, but it was its ancient ancestor, a simple strand, fragile, imperfect, but revolutionary. These early genetic molecules drifted inside the tiny lipid bubbles scattered across the tidal pools, and they behaved differently. When heat from the sun warmed the pools, the strands loosened and separated. When night fell and the water cooled, the strands folded back together, sometimes attracting new pieces, new matches, new partners. Copying. Not flawlessly, not consistently, but copying nonetheless. Every mistake became a variation. Every variation changed how that strand behaved. Some variants folded too chaotically and fell apart. Others folded in just the right way, stable enough to last a little longer, cling to minerals, gather food, or resist heat. Suddenly, molecules were competing, not consciously, not deliberately, but mathematically, chemically, inevitably. In the chaos of tidal cycles, some strands survived longer simply because their structures made them harder to destroy. Those longer-lived strands were copied more often, and with every accidental improvement, the chemical world took a tiny step away from randomness, and a tiny step toward life. Meanwhile, the bubbles, the primitive membranes, were evolving too. Some trapped genetic strands inside. Others collected simple energy sources, like charged minerals or reactive chemicals drifting in the pool. Those that held their shapes during heat and cooling cycles lasted longer and carried their molecular passengers into the next day. Membranes that stayed intact, strands that survived longer, reactions that copied faster. Piece by piece, molecules were building the first protocol of life. But something else was happening too. Something even stranger. Some of these early strands weren't just surviving. They were influencing the chemistry around them. A twist here, a fold there, and suddenly a molecule bent in a way that made nearby reactions easier. Some folded into shapes that helped bubbles form. Others helped molecules link together. They weren't alive, but they were starting to act like life. Tiny pockets of chemistry began to behave like miniature laboratories. Each bubble a world with different reactions, different strands, different destinies. Most of these microscopic experiments failed immediately, but a few survived long enough to try again, day after day, tide after tide, storm after storm. Earth wasn't just a planet anymore. It had become an engine, and deep in those tidal pools, something astounding was about to occur. The merging of two breakthroughs, a genetic molecule capable of copying and a membrane capable of protecting it. For the first time, information had shelter, and that single combination would start a chain reaction that would echo through every living thing that would ever exist. Life didn't appear in an instant. It assembled itself, one fragile piece at a time, and soon the next piece would begin to form, a piece that would change everything about how early chemistry survived, a primitive metabolism, the first flicker of energy flow, and with it, a new chapter in Earth's story. Life needed structure, life needed information, but life also needed something else, something just as important and far harder to achieve. Energy, because copying molecules, building membranes, repairing damage, none of it could happen without a steady flow of usable power. And in the early Earth, Power came from everywhere: sunlight, volcanoes, lightning, hot springs, minerals soaked in chemical gradients. But raw energy was chaotic, violent, destructive. For early molecules drifting in the tidal pools, it was more likely to tear them apart than help them survive. So the natural world had to invent a bridge, something that could transform wild, unpredictable forces into a steady chemical engine. And it began in the simplest possible way. Inside certain lipid bubbles, molecules that stuck to minerals began holding on to tiny electrical charges. Others captured reactive atoms drifting in the water. And inside those tiny containers, a pattern formed. Reactions didn't just happen at random anymore. Some pathways happened more easily. Others became more efficient. Some generated small bursts of usable energy, just enough to keep another reaction going. Primitive metabolism. Not life yet, but the shadow of life. The first flicker of organized energy flow on Earth. Tiny bubbles became chemical batteries. Some stored energy during the heat of the day. Others released it during the cold of the night. And in this endless cycle, the bubbles that used energy well survived longer than those that didn't. Inside them, the genetic strands, those early self-copying molecules, began to benefit from this stability. They lasted longer. They copied with more consistency. And in a few rare cases, they influenced the reactions around them. Some strands folded into shapes that accelerated the very chemistry they needed. Little catalysts, protoenzymes, molecular tools born from chance but preserved by survival. When a strand sped up a useful reaction, the bubble that housed it stayed intact longer. That bubble produced more copies of the strand. More copies meant more catalysis. More catalysis meant more survival. A feedback loop. The first spark of biological evolution. Nothing knew what it was doing, but the chemistry didn't need awareness. It only needed variation, and variation existed in abundance. Stormy days created violent reaction bursts. Calm days let fragile strands fold into new shapes. Tidal pools evaporated, concentrating molecules until reactions became unavoidable. Then rain returned, diluting the pools and forcing the survivors to spread. Every moment was a test, every molecule an experiment. And what succeeded because conditions allowed it. Slowly, the bubbles became more than shelters. They became ecosystems, miniature worlds of structure, information, and energy. The earliest ancestors of cells. They weren't alive, not fully, but they were more than lifeless. They were in transition, suspended perfectly between Earth's chemistry and the biology that was about to emerge. And they were almost ready for the next evolutionary breakthrough, a transformation far more dramatic than simple copying, membranes, or metabolism. A leap from chemical cooperation to chemical identity. The rise of the first true protocells. And when protocells took shape, the world would finally cross the boundary between pre-life chemistry and the origin of life itself. The moment was approaching, slowly, invisibly, over millions of years. A moment when the chaotic chemistry of the early Earth would cross a threshold and become something new, something persistent, something autonomous. The first protocells did not appear in a single place or at a single time. They emerged in countless pools, vents, cracks, and coastlines, each shaped by slightly different conditions. Some formed where volcanic minerals leaked into shallow water. Others formed at the edges of ancient tidal basins. Others still took shape where warm currents met cold ones in the planet's young oceans. Wherever they formed, they all shared the same basic ingredients: membranes that protected their internal chemistry, strands of molecules capable of copying themselves, and metabolic reactions that harvested energy from the world outside. But what made a protocell different from everything before it was not the ingredients. It was the integration. Inside these microscopic spheres, everything began working together. Membrane molecules reorganized themselves when damaged. Catalytic strands repaired other strands. Metabolic networks pumped energy into copying reactions, and copying reactions produced new catalysts that stabilized the metabolism. A circle, not perfect, not efficient, but a circle nonetheless, a closed loop of chemistry that could maintain itself long enough to matter. For the first time in Earth's history, a structure could support its own continued existence. The protocells that formed were simple beyond imagination. They could not sense. They could not move. They could not regulate temperature or control their environment. They existed entirely at the mercy of waves, sunlight, and luck. But they had something no molecule or membrane had ever possessed, a boundary between themselves and the rest of the world. Inside that boundary, chemical reactions favored stability. Outside it, the ocean remained wild, unstable, unpredictable. Every time a protocell survived a wave, a drought, or a burst of radiation, it carried with it a slightly improved arrangement of molecules, improvements that made the next generation of protocells a little more resilient. Evolution was no longer a passive chemical consequence. It was becoming a biological force. Some protocells developed stronger membranes. Some became better at absorbing energy-rich molecules from the water. Some copied their genetic strands more reliably. And some, by pure accident, divided into two smaller spheres when they grew too large. Division wasn't reproduction yet, not biologically. It was simply physics. But it created opportunity. Two bubbles meant twice the chance that at least one would drift into a favorable environment. Two bubbles meant twice the surface area to capture energy. Two bubbles meant twice the possibility that one would gain a catalytic mutation that made it more stable. And when division happened in protocells containing genetic molecules, the results became even more profound. Some strands were split unevenly. Some were lost entirely. Some were copied before division occurred. And occasionally, a protocell inherited a combination of strands that worked better together than the original. Natural selection had begun working on entire packages of chemistry, not isolated molecules. These were no longer random clusters. They were lineages. Lineages that could change. Lineages that could adapt. Lineages that could persist. The Earth was still violent. UV radiation still tore molecules apart. Tidal pools still dried into salt flats. Storms still swept millions of protocells away in a single surge. But life, primitive, fragile, molecular life, had found a way to continue. And with every cycle of destruction came new survivors, each carrying chemical arrangements that were slightly more robust, slightly more organized, slightly more capable of maintaining their internal order. This was the slow, patient birth of biology, not a sudden spark, not a miracle event, but a gradual drift from instability towards self-preservation. By the end of this era, hundreds of millions of years after the first building blocks appeared, the planet was home to countless protocell lineages. Some faded away, others left subtle imprints in the chemistry of the oceans, and a few, only a few, carried the combinations of structure, metabolism, and information that would evolve into the first true cells. Not yet, but soon. Soon the boundary between pre-life and life would be crossed completely. And when that happened, Earth's story would change forever. Because chemistry would no longer just react. It would struggle. It would compete. It would persist. And in that persistence, the earliest biological ancestors of everything that would ever walk, swim, fly, or think were beginning to take shape.